Our last speaker today is Donna Farber. She's familiar to most of you, I'm sure. Has a California connection. She did her PhD at the UC Santa Barbara and postdoc work at Yale and the Pasteur Institute before joining the faculty at University of Maryland, then moving to Columbia in 2010, where she is the George Humphreys Professor of Surgical Sciences, Professor of Microbiology and Immunology. And focus of her laboratory is on immune memory and immune responses in tissues. Her lab is well known for first identifying tissue-resident memory T cells that are in the lung and mediate optimal protective immunity to respiratory viruses. And it's great to have her today as our last and speaker. Thank you.
Okay. Okay, thanks very much. And thank you, Polly, for inviting me to participate in this symposium. It's been really a great day. I've just enjoyed all the talks, all the range of what we need to know, right, about vaccination. So I'm going to be talking about immune memory to vaccination across tissues and age. And as all of you know, immune memory is maintained in multiple tissues. And it's initiated in lymph nodes, but it's maintained in other lymphoid organs throughout our body, as well as barrier sites such as lungs, intestines, and skin, and really virtually any tissue you can think of, like even the liver, exocrine sites, pancreas, will have memory T cells in particular. And why that's important is that where a T cell is very much linked to their protection. So as mentioned, tissue-resident memory in barrier sites such as the lung mediate optimal protection to all sorts of respiratory viruses. So in the lung, they'll mediate optimal protection to flu and RSV, mycobacteria, virtually any pathogen that's been looked at in mouse models. It's those tissue-resident cells that are already in the lung that are maintained there and adapt to that tissue that mediate that great protection. And the same is true of the skin, of the gut. But then you also may need, and these resident memory have certain phenotypes that distinguish them. They're also, they tend to be in certain tissue niches. They express CD69 and some other integrins. But there's also different types of surveilling memory cells, such as central and effector memory cells. And those are required for protection to more systemic pathogens. So where a T cell is and how it migrates is really critical for protection. But it's not just T cells, but B cells are also maintained. Memory B cells are also maintained in tissues. So they're maintained mostly in lymphoid organs, different kinds of memory subsets. And then, of course, plasma cells are not only maintained in like the bone marrow, but they're also major subsets in intestines. So a lot of our plasma cells, particularly in humans, most of them are in the gut. And so we talk about, antibodies and titers and how long that takes, how the half-life of a titer in terms of vaccine efficacy, but it's really the cellular stores of memory that are in tissues that are going to replenish. Those are the source of those titers, whether they're memory cells or plasma cells. So it's important to understand where those memory B cells are, and that would be important for a type of vaccine. So that's what I'll be talking about is our studies in human tissues and trying to understand this question. So we know there's many different kinds of vaccines. And of course, the childhood vaccines are really some of the most successful vaccines: measles, mumps, rubella, diphtheria, tetanus. All these vaccines that we get in childhood are, give you great, they're great protection. They give you very long-lasting antibody titers. And, despite what's coming out of Washington, they're also really safe. And we know that there's different kinds, there's different formulations, are given mostly injectable, but there are different routes. And then we also know that there's like shorter lived memory or memory that's, the vaccines are not as fully efficacious as we might like in terms of protecting from actually getting any sort of illness at all. I mean, there's different levels of protection, as we heard in the morning, protection from death, protection of serious disease, but not necessarily protection from, the sterilizing immunity that we want. So what I'll talk to you is about somewhere in our studies and how we're trying to understand in humans how this vaccine-induced immune memory across tissues compares to circulation. And this is really the dynamics and magnitude and different functions. And then what is the role of the tissue, of the antigen? and age as well, because age is always a big factor, right, in any human studies, in how this is maintained. And so what we're using to address this in humans is this tissue resource that we set up over 15 years ago now, where we get tissues from human organ donors. And the way we do this is that we actually have this long-standing collaboration with our local organ procurement organization in New York City, in the New York metropolitan area. And we have on-call surgeons in my lab that go out and get and can get these tissues. So we're sort of we go there at the same time that the tissues are obtained for life-saving transplantation. And these are all donors whose next of kin have consented for the use of tissues, not only for transplantation, but also for research. So, so we've been doing this now for 15 years, and we've gotten tissues from over 850 donors at this point. We have several resources. One is we also have access to pediatric tissues through interactions with organ procurement organizations across the entire US. And there's several advantages to getting tissue from organ donors. One is that we can get many tissues from one person. So we often get all of these tissues here, lungs, intestines, all sorts of lymph nodes that are draining each of these different tissues, and then primary and secondary lymphoid organs like the bone marrow and spleen. And then we also get some other sites, such as skin. I'll be talking mostly about mucosal lymphoid sites, bone marrow, and blood. And so we can do that, and we get these fresh, so we can do functional studies and other profiling like site-seq. And then the other big advantage is that we get donors of all ages of human life. And as we keep accumulating this and adding to our bank, we can then do studies where we're looking really across tissue and across age for most every parameter that we're looking at. And this just shows you, we also get a lot of information about the donors because they do a lot of serology. They want to make sure these donors are not going to have an ongoing infection or chronic infection. The donors, meet a criteria of health, because remember, their organs are going into immunosuppressed people who are critically ill. So, they don't have cancer, for example, so they have a certain criteria of health, which is basically about people walking around, sort of that level of health. They're highly diverse. And we get the stereostatus EBV, CBV. And so we're using these to address this question. So we've done a lot of profiling, and I'm a T-cell biologist by origin, at least from my postdoc on. So we've done a lot of profiling of T-cells, but we're broadening out. And so this is a site-seq profiling. This is sort of our most broad and kind of detailed in terms of single cell profile to date. And it just came out, it's out like last month. And this was done with all the major immune lineages aside from granulocytes, which you need to remove from tissues or it kills everybody, from 24 donors. It was a consortium with the UK, so 12 were from New York, 12 were from UK. And it was in collaboration with Peter Sims in system biology at Columbia, as well as Nir Yosef, who I know is a collaborator with some other people here from the Weizmann Institute. And this is a site-seq, and we did both protein and transcriptome profile. We got 1.2 million immune cells, and this from all these tissues: blood, bone marrow, spleen, lung, guts, and those were the major sites. And you can see we get the major lineages, and this is a UMAP of the whole data set, so that the clustering, the segregation is really by immune lineage, okay? But then when you do for each lineage, and if you look at the variability in the data, and this is a variance decomposition now. So you ask, where's the source of variability? For each lineage, the source of variability between these 24 diverse donors on either side of the Atlantic Ocean is tissue. It's not donor, okay? So for CD4, CD8, NK, and myeloid, it's really the tissue that has a major role in this immune cell identity that doesn't really differ between people, okay? And then we did, just to show you there, we had over 500,000 T cells and hundreds of thousands of B cells. And when you can look at them, we did a classifier where we integrate both surface protein and transcriptome profiling, to more accurately annotate these cells. And you can see that you get these tissue resident memory here, and that kind of segregates with the gut. The gut has very strong tissue adaptation, and the lung too, you can't really see it on the UMAP. Memory B cells are mostly in these lymphoid organs here. And you get also plasma cells, and you can superimpose with the tissue as well. But one other feature of the tissue is not only that they differ between the lineages, but it's also the composition of immune cells. It's a feature of the tissue. Again, something really invariant. And this is again showing now as proportions of all these CD45 positive immune cells from blood, bone marrow, spleen, lymph nodes, lung, and intestines. And just sort of highlighting, you can see that the composition, and this is again from 24 donors, so it's very consistent across donors. And most of this we validated with hundreds of donors by flow, especially for T cells. But it's really the kind of the most the frequency populations, like for T cells, for example, it's really a feature of the tissue. So you see in intestines, the major T cell population is TRM, is these tissue resident memory, okay? And whereas in the lungs, you also find resident memory, but you also find some circulating memory in the lungs. Whereas a lymph node, you have more central memory, and you have regulatory cells, and you also have resident memory. Whereas in the blood, you don't have resident memory, you have these naive and surveilling populations, okay? So you can see that the blood is quite different in composition to all of these tissues. And then for B cells, what's interesting about B cells is that B cells of the major population of B cells in the blood are naive, right? That's the major population of naive B cells. Whereas memory cells, their major, the major reservoirs are in lymphoid organs, spleen and lymph nodes, okay? And a little bit in bone marrow, okay? Whereas plasma cells, you get the high frequency in gut. You also find them in lymph nodes. And then they're also found in bone marrow, but it's actually really, they're kind of relative, they're kind of a low frequency relative to the other immune cells. So these compositions are very tissue specific, okay? So now we want to go in and look at, well, what about the antigen-specific populations? Are they simply a feature of the tissue, or are there changes in the type of antigen you use? So when we first started looking at antigen-specific T cells, we were relying, it's kind of tricky, of course, in humans, because you need to rely on these multimer reagents. You need to know the HLA type of these donors, which we do. And so we looked at, because these are donors, we just get one look at them. We started to look at ubiquitous pathogens such as flu and CMV, and we would use, this is tissues across one person. These are just 3 different lymph nodes: lung lymph node, iliac lymph node, and mesenteric lymph nodes. And we could look at either where were the flu-specific CD8s, and in this donor, most of them were in the lung and the lung-associated lymph node, whereas the cytomegalovirus-specific CD8s had a different distribution. There were a lot in the bone marrow. They were really kind of disseminated across sites. When we looked at what are the types of T cells that were stored, we found that to the flu, to influenza, many of these were tissue resident memory phenotype in the lung, the spleen, and the lymph node. Whereas for CMV, we didn't find really tissue resident memory. They're more surveilling. And it was also the kind of the tissue distribution was different as well. So we can see from an infection, where you're storing memory really depends on the type of virus. But what about vaccines? So the pandemic, of course, gave the opportunity to look at where we could distinguish between a vaccination and infection. And we can actually look at the timing, because we could look up when they were vaccinated in the New York State Registry. So this gave us a unique opportunity then to look at tissue immunity by using organ donors to look at tissue immunity to a vaccine or a vaccination infected. So during the first 2 years of the pandemic, we acquired a cohort of about 20 organ donors who were vaccinated but weren't infected. And then, of course, most everybody now is vaccinated and infected, and probably many times. Then we had pre-pandemic controls. You can see that we're going across age for all of these different cohorts. They all have antibodies. So the vaccinated cohorts had anti-spike antibody. They had RBD, sort of the neutralizing sort of domain. And then we also did the microneutralization assay. And what you can see in this sort of little person plot, this is just sort of the frequency that had neutralizing activity, that it was much higher in the donors that were vaccinated and infected than the vaccinated alone. But we can measure some neutralizing activity. So they all had, antibody memory. So when we looked at where the memory was, and this was for T cells, we didn't need to do HLA type because of the AIM assay, the activation induced marker assay, which Shane Karate and Alex Sette of the Hoya Institute developed. And in fact, Alex Sette gave us the peptide pools of spike to use for our assays. And so what you do is you take the mononuclear cells from all these different sites, you incubate them with spike peptides, and then about overnight, the next day, you look for induction of multiple early activation markers like OX40 and 41BB. And what you can see from the vaccinated unaffected cohort, is just a vaccinated cohort, you can see responses in many sites. You can see them in blood, bone marrow, in the lung, and a little bit, most in the lung lymph node, not so much in the mesenteric lymph node, but not in the uninfected control. And what you can see is that for the CD4s, this is really a CD4-driven response to spike. With the AMAS, you get much more of a CD4 response. And the magnitude of this response across tissues didn't really differ between the vaccinated or the vaccine infected. But what was kind of interesting is that when you look at the frequency of the donors that had a measurable response in these different across tissues, the most consistent response was in the lymph node and in the lung-associated lymph node. The lung-associated lymph node in humans is always a little bit reactive. It's like the biggest lymph node that we get among the lymph nodes that we get. And so there was a very consistent response there, and in some of the other lymph nodes, as well as the spleen, a little bit less in the lung. But what you can see is that we didn't measure the response in the blood of everybody, but we did measure the response in the lymph node of all the donors that we did. The response to CDH was much lower. But we could also find it in the lymph, mostly in the lymphoid organs. It was mostly an effector memory, as shown here, with some central memory in the lymph nodes. And we did look at tissue resident markers, also using multimers, and they were pretty much 50 to 80% CD69, which is a canonical marker in tissues, not in the blood. And they also, many of them express CXCR6 in the spleen, lung, and lymph node, even with the vaccinated. So you are getting some resident markers that are induced with the vaccine. But in terms of the dynamics, if you look across age or across time post-vaccination, that was different in tissues over blood. So this is showing the relative memory CD4s in tissues over blood or minus blood. And what you can see is that over age, you're more likely to find this response in the spleen compared to blood or in lymph nodes compared to blood. So it might disappear in the blood, but you could also find it in older people more often in lymphoid tissue. So it doesn't mean so, and that's actually important to know because it means, a lot of times it's for older individuals, they recommend more boosting because you tend to, not be able to detect the response in blood, but it doesn't mean necessarily that they don't have a memory response because it's actually in the tissues. And the same thing for time post-vaccination. Again, it's known that even T cell response in blood does wane over time post-vaccination, but it didn't wane in the spleen. And you were more likely to see it in the spleen than the blood, suggesting spleen may be a reservoir for long-lived memory T cells, like it is for B cells. Yeah. So it's just looking at the dynamics and looking at the tissues gives you sort of a different view in that, what's happening in blood may not be reflective of what's happening in these lymphoid organs. But then what we found that was a bit surprising, though, when we started to delve into what are the functions of these vaccine reactive T cells, these spike reactive T cells in different tissues, was that we found that a lot of them were regulatory T cells. So regulatory T cells are defined by CD25 and FOXP3, and of course, the Nobel Prize just awarded for the discovery of FOXP3 and Tregs by Sakaguchi and Fred Ramsdell and others. And what you can see is that regulatory T cells, either in the blood or lymph node, they're very low frequency. It's about 1 to 8% in the blood. It's a bit higher in the lymph node. But when you gated on the spike reactive cells, or the AIM positive cells, you can see that about a third of them were Tregs in the blood, and more than half of them were Tregs in the lymph nodes. So this was kind of surprising. So we looked at many donors, and what you can see is that there are high frequencies of the spike reactive cells that are Tregs, particularly in the spleen and in the other lymphoid organs, less so in the bone marrow, and it's really variable in the blood and the lung. But we were definitely finding Tregs. So of course, and it was in the vaccinated donor or the vaccinated infected. So of course, first time we found that we really worried. It's like, oh, is this an artifact? Because FOXP3 can be induced from activation. So we asked, well, maybe this is. So then we activated. So what we did is we took these T cells from the different sites, we activated them polyclonal with anti-CD3 and anti-CD28, and then we sorted those AIM positive cells and we looked for FOXP3. And this is showing you sort of the paired analysis of the spike reactive versus the polyclonal active. And what you can see is that it was enriched for every single site. It was really enriched in the spike reactive, and it was not due solely to the activation of the cells. So we're clearly getting some enrichment. And so we wanted to look closer into this. Was this just with spike, or was this also other antigen-specific cells? And so what we did is we also did site-seq of the AIM-positive cells that we did not only with spike from the different sites, but also with influenza peptides. And we did find the same thing. In fact, that these AIM either spike or flu, which is F here. And this is just showing you sort of the proportion of different cells. The orange are CD4 and sort of the melon color, the light orange, are Tregs. That this is sort of the non-activated composition of CD4 and CD8 cells. But you can see it's enriched in the blood, and it's very enriched in the lung and the lymph nodes. So even for flu. Now, this also shows you the entire T cell response, including CD8. And you can see in the bone marrow, it's mostly CD8 driven for both spike and flu. These are CD8 effector memory and temora cells. These are effector-like, and it's a little less enriched in the spleen. So there's certain tissues where you find this enrichment of Tregs for memory cells, okay? Memory CD4, and they are memory. So if you look, here is a site-seq. You see it's CD25 and it's CD45 RA low. So these are really like a TEM phenotype for memory. So we think that this means that there are like a significant proportion of regulatory cells that we're generating in lymph nodes to either vaccines and also to some infections. And I will tell you that since this was published at the end of last year, I've had a lot of people come up to me that they are finding the same thing to other antigens. So regulatory memory in humans is a real thing, okay? The question is, what does it mean? So we know that, well, tissue memory cells are, so just to summarize, tissue memories are more durable, maintained with age compared to circulating counterparts. They may be even preferentially generated with age, these tissue. And that we're generating these memory Tregs. The question is why? You know, we sort of think of, well, when you want memory, you want protection, you want to have pro-inflammatory Th1, you want to give gamma, you want to get rid of the virus, right? I mean, that's true. But on the other hand, if you have too much of that, that's bad, right? And we found in COVID, what happened when nobody had any memory T cells to COVID? You had this massive lung infiltration and nothing to regulate it. And it could be that over our lifetimes, you know, as we keep encountering pathogens, you know, as adults, we really don't need that strong inflammatory response. What we need is to regulate everything. So that memory Tregs could be positive. Now, they also could, you know, act to limit tissue damage. And that's why we might be seeing them more in tissues than certain lymphoid organs and in circulation. They also could be, on their hand, it could be bad in some cases. So maybe the regulatory T cells are limiting the antibodies. So when you have them to spike, maybe that's not a good thing. And we've done more donors and are trying to do those correlations with antibody to see if you have more Tregs in the site, do you have less antibody? So I think there's a lot of questions there. But again, for vaccines, what do you want to generate? You probably want to generate the right combination of CD8 like effector and a nice regulation so you get that balanced response. Okay. But then we wanted to look at other vaccines. So we know that, here we have a vaccine, SARS and flu, we know it's not as long lasting as some of these other vaccines. And so we really tried with measles and mumps, those are really durable vaccines, to look at that peptide, to get peptides with Alex made us and try to get a T cell response. It was just so hard to even find any response. And so we thought, and we wanted to go across of these different antigens. So we decided to go with B cells. And you've just heard from Scott, we didn't have this, it's the same technology that he has, but we actually are going to collaborate with him in his really super technology with all the different specificities. But what we were able to do, and we did this with SARS too, as well as do those, we have the biotinolate and you derivatize and you make tetramers. And we were able to get some good proteins because we have a measles virologist down the hall, to get these proteins and then validate these. And with B cells, it's really beautiful. You do not need to know the HLA type. You just take the protein and you pick it out. And so what we did is we looked for and to compare between these antigens: measles, mumps, chickenpox, tetanus, and diphtheria for subunit vaccines. Then we did flu and SARS. And we did this now from 107 donors. This is a project of Eric Zhang in the lab. He's an MD PhD student, and across age. And so we're really looking mostly in lymphoid organs. That's where most of the B cells are, different lymph nodes and spleen. And so just to show you that we can identify, and these were also validated with monoclonal antibodies as well, these probes that we can look at multiple, we can detect all of these proteins. This is a spleen from one person, and they're in the memory fraction and not the naive. So that's also good. And then if we look across now, all of this is all the data, we're looking across blood, bone marrow, spleen, and the lymph nodes. And what we find just overall is that we can find it across all these tissues. We can find these memory B cells specific for vaccine antigen. But there's a couple of take home here, and I know it's complicated, is that one, across the board, we get the lowest response in the mesenteric lymph node. And it's actually really hard to find memory T cells in the gut. So we think that when you're vaccinating, you're really not vaccinating for gut immunity when you're doing a systemic vaccination, okay? It's higher for SARS, but that's probably because it's more recent. And so that's sort of the overall, but we are finding it everywhere. Now, what about the subsets? So we're really interested in memory subsets. And, this is newer to me about B cell subsets, but you can identify B cell subsets by IgD and CD27. These are the naive cells. And then you have classical memory B cells, which are CD27 positive. They can be class switched or not. And then atypical memory B cells. And these were really identified by the late Mike Cancro, and others have worked on this in mice as well. It's in mice and humans. These are CD27 negative, CD21 negative memory cells. Are not as functional, and they're also called age-associated. They're not as functional as a classical. And so we could find that classical memory B cell, this is just polyclonal, or mostly in lymphoid organs, and atypicals are in bone marrow. So then we wanted to see, well, of these antigen-specific cells, what kind of subsets were they? And what you can see, and again, I know there's a lot here, this is class switch memory and non-class switch memory. So I'm just showing now the classical memory. You can see it's higher in the spleen and lymph nodes across the board, but there's a couple of differences. One is that there are some non-class switch memory enriched in blood for certain specificities, and particularly for tetanus toxoid. There's a lot of non-class switch memory across tissues for tetanus toxoid. We don't know why. There's all the SARS and flu are mostly class switch. And then you can look at isotype as well. And again, SARS and flu is practically all IgG. You're seeing these multiply boosted, whereas for tetanus toxoid, you have more IgM. And for some of these other, but for measles, again, it's mostly class switch. But then there's two other these interesting subsets. One is the atypical or the age-associated, and then also you can have resident memory B cells. But the atypical are interesting because they're mostly found in bone marrow. And you don't find that many of them for the vaccine antigens, for the childhood vaccines antigens, measles, mumps, or a little bit for chickenpox. But you do find them for SARS, and particularly in the bone marrow and spleen, and also for flu in the bone marrow and spleen. So where you're getting the atypical are from sort of the ubiquitous pathogens. Where you get residents are mostly in the lymph nodes, or practically all residents in the spleen. And what's interesting about these subsets is they change with age. So you get more atypicals with age to SARS and to flu in the bone marrow and the spleen. Now for SARS, this is not maintenance necessarily, because these people were probably getting immunized at that age. So it could be the later you're immunized, the older you are, the more you're likely to make atypical memory. Okay, so, and those are not as functional. You tend to accumulate over as these memory cells are maintained, they become more resident. I know it's a little complicated here, but just showing that measles, tetanus, they all in the spleen and the lymph nodes, you accumulate, you become more tissue resident as you get older. And so finally, so what is the source of these different subsets? So we wanted to look at first the subsets with more resolution using single cell RNA-seq, and then also do simultaneous BCR clonal analysis to see if these different subsets were arising from different clones going to different sites. And so what we found, sort of the major finding from looking at these memory B cells, and this is 3 donors, and we did 4 different antigens that we sorted for, is that they do segregate by tissue, but they also segregate by different kind of memory subset. And we found classical and atypical memory here in the blue, but we also found, and this was because of the resolution of single cell, that we had also a lot of marginal zones memory cells. And marginal zone they arise from transitional B cells. They tend to be more in the spleen. In a mouse, they're only in the spleen. In human, they can be multiple lymphoid organs. So we found marginal zones, and here's just some of the genes, the canonical genes, and we also validate this by flow that we are getting marginal zone cells. And the proportion of these subsets differs between sites for the different antigens. So you can see in the youngest donor, the green is marginal zone. There's a lot in the spleen. And you can see the atypical is enriched in the bone marrow for SARS and flu, very much like the flow, and that there's more atypical in sort of the oldest donor. But when we look at the clones, that was also interesting, because what we find with the clones is that we get very highly expanded clones for measles-specific B cells as well as tetanus-specific B cells, less for SARS and flu. And that was really across all 3 donors, okay? And just to end up, because I know it's getting late, so then we wanted to know, well, are those clones, those expanded clones, are they found in single tissues? Are they across tissues? And what's the ICE type and the subset? Because again, that question is how this distribution across tissues are regulated. Is it different clones? Well, it turns out it's not different clones. And if you look at the top expanded clones for all the different specificities, that you can find that you get these clones that are disseminated across the 3 sites. When you look at ICE type, it tends to be pretty constant that one clone that is expanded and overlap has a similar ICE type. So these are all mostly IgG. Here's an IgA clone for measles, and here's some non-IgM. For tetanus, as I said, they're really expanded, but they're non-class switch, and I don't know why that is. I think that's really interesting. And then SARS, it's sort of, again, you get some overlap, and the ICE type is really, it's really a feature of that antigen, the antigen. But when you look at the subsets, it's different. Then it becomes about the tissue. So if you look at the Hispanic clone here for measles, you can see that it's enriched in marginal zone cells in the spleen, in classical memory in the lymph node, and in atypical in the bone marrow. So one clone becomes 3 different identities depending on what tissue it lands in, okay? And it's the same thing here. You can look also for tetanus. You have marginal zone in the spleen, you have class switch in the bone marrow. And you can kind of look, here's more atypical here for measles in the bone marrow class switch. So it seems like, and here even for SARS, here it's marginal zone in the spleen, class switch in the bone marrow. Here's one that's atypical in the bone marrow, overlap with the lymph node. And then for the flu, most of the expanded clones were in the spleen, but again, it's that tissue-specific subset. So just to summarize this last part, is that B-cell memory to childhood vaccines is stably maintained in lymphoid organs. So I think that's something that we found, and that was really nice to see. And I didn't show you, but it actually correlates with the serology. The memory B cells, mostly in lymph nodes and spleen, really correlates with the degree with the antibody. And that the subset is a feature of the tissue for a given clone, while antigen drives the ICE type. Okay, so there's some regulation with the tissue, and we don't know, do they land in that, or is it really the tissue identity that causes, as the memory cells are being maintained, to have that identity? We don't know that. There's a lot of questions here. And that changes in class switching and increase in residency and atypical memory, they change with age. That's another consideration. So again, these considerations about how do we want to direct memory? How do we want to vaccinate to direct our memory B cells to certain sites? What's the best site for them to be in? And one might think, well, maybe it's best really to be a class switch memory B cell in a lymph node, because you're just going to get better and better and more resident. But we'll have to see. So I just want to thank everyone who did the work. This is a big team effort. I want to shout out to my collaborators, Peter Sims in systems biology, and also in pediatrics, Matteo for all of the measles and mumps antigens, and then our collaborators at La Jolla Institute with the SETI lab, also at Scripps, and of course our funding and the donors. In fact. So thanks very much.
Questions?
Thank you. Wonderful talk. So I have a question about the Tregs, these memory Tregs. So do you think that I understand you think about these cells as bona fide Tregs that are there to prevent excessive inflammation, but do you think that under the right circumstances these cells can become effector cells upon antigen exposure?
Oh, when you activate them, it's a really good question. Well, what we found, and they're a little bit unusual. So they have all of the canonical transcriptional and protein features of Tregs. I mean, everything, CTLA4, TIG, all of that. But when you activate them, they produce the chemokine CCL17, which is produced by no other human T cell anywhere that we've looked. And they also express CCR8. So we're looking into those features of it, and especially what this chemokine is doing, because we think maybe it has some kind of weird function. It's just very unusual. They're not really producing IL-10, so they're not sort of like mouse Tregs in that way. Yeah, so but this is what we're looking into more. Yeah.
Wonderful talk. I had a question if whether you had a hypothesis around whether it's a feature of the antigen being presented in the vaccine or the vaccine formulation that's driving those B cell phenotypes and isotypes that you're seeing and tissue distribution.
Yeah, I mean, I think it's definitely the antigen. I mean, the most curious one is the tetanus and diphtheria, because it's completely different subsets. Like tetanus is not class switch, and you're getting them at the same time, right? It's DTaP, right? So you're getting them at the same time, same formulation. So that's like a good control. Same with like measles and mumps, but yet they're very different. So I think that it's definitely the antigen, maybe how it's presented. I think T cells have a lot to do with it. So I wonder if maybe they're not switched because you're not getting the right kind of T cell help for some reason. But I think it's probably that the antigen is playing a lot at how it's being presented.
Thank you.
And presumably both the diphtheria and the tetanus are not really being boosted down the road post-vaccination.
No, you wouldn't think that. I mean, you usually get a boost every 10 years of tetanus, but not diphtheria. It's the tetanus boost you get.
Yeah.
But even it doesn't account for with the boost.
Thank you for very extensive work. I have some concerns about possible selection bias on cause of death of the donor and prior history of the donor use of antibiotics or antivirals that could be affecting your distribution and type of B and T cells.
I mean, so we have done stratification by cause of death. So these are all traumatic, non-infectious causes. So it would be, like a suicide, a homicide, some sort of motor vehicle accident, cerebral vascular stroke, or cardiac event. Usually in anybody like in the 40s or above, it's about cerebral vascular stroke, an aneurysm.
So they're particularly my concern because cardiovascular deaths make up a large percentage of our deaths in this country. That's an inflammatory type effect. And
yeah,
so
right,
I'm just wondering if you have that data. Prove me wrong. Say no, it doesn't make a difference. People who got antibiotics or didn't.
Well, antibiotics, we don't have that information. So that's just, you can't really control for it. I mean, but as much as we control for that, we do stratify everything by age, by sex, by BMI, and we've done it by cause of death with multivariate models for all of this. And this is why we use hundreds of donors. And that's why and actually our data are very robust for that. That even, for all these covariants of people, and when we do the multivariate models, it doesn't come up. Age will come up and tissue comes up.
Very exciting data.
Other questions? Okay, one more.
Do you think that the T cells, the memory T cells, just sit there in that organ forever? You don't think they travel and survey?
I think they may be moving around in the tissue. We've done a lot of TCR sequencing, and we can find clones that are only in the gut or in the lung that are TRM, and we don't find them elsewhere. We find more overlap with the associated lymph nodes. So like there is overlap of the gut and the gut-associated lymph node. But you also find clones that are really enriched. So we think that they are staying there.
Yeah.
Yeah, and certainly in transplantation, like in lung transplantation or gut, you find like donor cells that come in with the transplant. They just sort of hang out there. They don't go out.
All right, let's thank Donna once more.
Mhm.